So in this video, I'm going to go over the whiteboard drawing that I did in class. So everything starts with the compound glucose, and glucose undergoes a process called glycolysis. And sorry, I spelled it wrong. Glycolysis. Oh gosh, that's spelled horribly wrong. Oh, that's not too bad. So it undergoes a process called glycolysis. And during glycolysis, there is the need to have NAD+. And what is made from glycolysis is a little bit of ATP and NADH. The formation of ATP during glycolysis is called substrate level phosphorylation. And phosphorylation, remember, is just the addition of a phosphate to ADP, so that is our energy storage molecule. Now, after we take glucose and it undergoes glycolysis, what it ends up being is pyruvate. At that point, we have the decision as a bacteria whether or not you are an organism that lives and uses oxygen, or whether or not you're one that does not use oxygen. If you do not use oxygen, then this pyruvate is going to undergo the process of fermentation. The process of fermentation is merely so that we can make more NAD+, so that glycolysis can then happen again, and we can get another little bit of ATP. I think it's something like 2 ATPs, but again, I'm not really concerned with the numbers. As a byproduct of fermentation, either alcohol or lactic acid can be made. So we have acidic or alcoholic fermentation, and these are byproducts that we use like for making beer or something like that. And remember that this entire process requires no oxygen. So no oxygen is needed for this process. Okay? Now, let's say you are an organism that wants to use oxygen. So this pyruvate then is going to undergo the Krebs cycle. And when the Krebs cycle happens, we get a few more things that come out of the Krebs cycle. We get a little bit more ATP. We get FADH2, and we get some more NADHs. Okay? Now, what happens with these things that have H's on them? So we've got NADH and we have FADH. Let me get another piece of paper here so we can see all that. What we have made these for is essentially so that we can have the hydrogens. Okay? So the FADH, the NADH, the NADH here are going to make their way to the cytoplasmic membrane, where there is an enzyme, and this is called the proton pump, or the hydrogen pump, it's also called. And remember, this is in the cytoplasmic membrane of the bacteria. So let me just kind of draw this. And then we have this big enzyme. It's usually drawn like this. Okay? So, along come the FADH here, and the NADHs come over here, and what ends up happening is the proton pump takes a hydrogen, with hydrogen as number 1 on the periodic table, which means it has 1 proton in its nucleus, and it has 1 electron on the outside. So this is a hydrogen atom. So what it does is it rips that hydrogen apart, and it pumps the proton out here. So this is the cell wall, the layer of peptidoglycan, and so the protons get pumped out here, and the electron from each hydrogen atom gets passed down these things called cytochromes. This is the electron transport chain. So the electron transport chain is this whole process here. And this is inside the cell here, and this is outside the cell. Okay? So these protons here are building up on one side outside the, so we get like a lot of protons out here. And so it essentially builds up this electrical gradient. And there's some electrons down here, and so the protons are attracted to the electrons because the oppositely charged particles attract one another. And you have some oxygen sitting down here, which we call the terminal electron acceptor. So the electron comes and jumps off of the cytochrome, and a proton comes in and is attracted and attaches here. And when those electrons come back together, they are a hydrogen. So this happens 2 times so that you hook up with these oxygens and you end up with H2O2, which is hydrogen peroxide. As the protons rush through this enzyme is ATP synthase. And when this happens, we make a huge amount of ATPs. I think it's like 32 ATPs. I don't really know the exact number, but it's a huge amount of ATPs. This process right here is called oxidative phosphorylation. So this is oxidative phosphorylation. Over here, where we make a couple ATPs with glycolysis, is substrate level phosphorylation. So hydrogen peroxide is, again, considered one of those toxic oxygen species. So this is toxic oxygen. So the bacteria need to have the enzyme catalase present so that they can break that oxygen down into water, which is not harmful, and oxygen, which is not harmful. Okay? Now, what else can happen is over here you've got all of these oxygens sort of hanging out, and if instead the protons and the electrons come back together and form, the hydrogen peroxide, sometimes an electron accidentally jumps off here and ends up charging up this oxygen molecule. This right here is a superoxide anion, another form of toxic oxygen, which is dangerous and sort of just accidentally happens when we're using oxygen as our terminal electron acceptor here in the electron transport chain. So in order to cure us of the problem of superoxide anion, we need the enzyme superoxide dismutase, and that turns it into water and oxygen, which is not harmful. Okay, so this whole process right here, going all the way from glycolysis through the electron transport chain, is considered aerobic respiration. Okay, so let me just grab one more picture. Okay, so we have glycolysis. I'm just going to shorten it up here. Glycolysis, and then we have fermentation. Okay, or from glycolysis instead we could go this way and we could do Krebs cycle, Krebs to the electron transport chain. Okay, so this process, these right here, this is considered aerobic respiration. Okay, now we do have the case where we have anaerobic respiration. So I guess I should add here that O2 is at the end. Oxygen is at the end of the electron transport chain for aerobic respiration. Some organisms can do Krebs cycle, have the electron transport chain, and then have like nitrate or sulfate, something other than oxygen at the end. This process is called anaerobic respiration because it does not involve oxygen and doesn't have the trouble that we have with these toxic oxygen species. Okay, so we have aerobic respiration where you make a ton of ATP. We have anaerobic respiration where you also make a lot of ATP, we just don't have oxygen at the end. Or we have this process of going through fermentation, which makes very small amounts of ATP. And this is considered an anaerobic process because there is no oxygen present. However, it is not respiration, which is this step that goes through the Krebs cycle and the electron transport chain. So hopefully this has helped to re-explain these ideas and it is clear to you now.